Dogma is a process that begins with DNA, moves to RNA, and finally to protein. Every single living cell in every single organism has to complete this process. Like this fish, this blade of grass, this banana, even a little paw has to do it. What is DNA exactly? Well, it stands for deoxyribonucleic acid, and it's a tiny, tiny molecule that holds the genetic instructions to make all of our proteins that our body needs. It's shaped like a double helix, made of a sugar phosphate backbone and a combination of 4 nucleotides: adenine, cytosine, guanine, and thymine. A matches with T, and C matches with G. So the first step is called transcription. This converts the DNA to the RNA. The specific part of the DNA that is used to make RNA is called a transcription unit, and where that section begins is called the promoter. This promoter includes the top box. RNA polymerase unzips the DNA and starts to add nucleotides that match with those on the template strand to make the mRNA. In RNA, instead of thymine, uracil is matched with A. When the RNA polymerase reaches the end of the transcription unit, it releases the RNA transcript, a process called termination. Now that we have the mRNA, it's on to the next step, translation. Translation occurs in the cytoplasm after mRNA moves from the nucleus to there. In this process, the mRNA is fed through a ribosome, which is made up of proteins and ribosomal RNA. The purpose of RNA is that it has binding sites where the transfer RNA can interact with the mRNA. The tRNA consists of an anticodon at one end and an amino acid on the other. The ribosome begins reading the mRNA at the start codon, AUG. The start codon then determines the reading frame that separates the nucleotides into triplets called codons. The anticodons on the tRNA match up with the codons on the mRNA, which codes for a specific amino acid. The amino acids on the other end of that tRNA begin to chain together in a polypeptide chain, also known as a protein. Eventually, the ribosome will read a stop codon. This signals the end of the protein. This protein could have many different uses. For instance, it could be an enzyme that catalyzes a reaction.